This is a tutorial for how to use Merizo on the Cypred web server. Merizo is a tool for automatic protein domain segmentation. So given a multi-domain protein structure, Merizo will attempt to segment that into domains. Merizo is a deep learning based method that does automatic segmentation, and it is trained to produce domains that resemble those found in the a.k.a. database of protein domains. Because it operates on protein structure, the first step in job submission is to select the PDB structure data tab and select Merizo as the analysis. The next thing you need is a PDB file to segment. I've collected a few, so I'm going to select one, this one. And then here on the right hand side, we can set some options. If your PDB file has multiple chains in it, you can select which chain you want to analyze. In my case, I'm interested in chain A from this file. It's also important to note that your structure does have to be in PDB format rather than MMSIF. MMSIF support is hopefully something we'll be adding soon. After you've selected the chain to analyze, you can choose one of two segmentation modes, default or iterative. Iterative simply means that Merizos run again on the result of one pass of the method to try and refine the result. And it's most useful when your chain is approximately 800 residues or longer. It is also useful when you're dealing with models taken from the AlphaFold protein structure database. In my case, this protein is roughly just under 700 residues long, so I'm going to stick with default because it should be okay. If I'm unhappy with the segmentation result, I can always go back and run another job with iterative selected. So I'll just say test. You can give your job a useful name, and then I will write a helpful name, and optionally you can specify an email address where you'd like the results to be sent. And then you click submit. For small to moderate size structures, Merizo does run quite quickly, so your results should appear fairly soon. Okay, our result is ready. So we'll go through the different parts of this results page. Up here where my mouse is, we have a sort of display viewport that shows the segmented structure, and the individual domains are given different colors. And so we can spin the structure around and take a look and see whether we think the domain segmentation is believable. In this case, this structure appears to be comprised of a large Tim barrel sort of domain, and then three domains that look like they're immunoglobulin-like domains. I'm not particularly sure about this last one, but yes, they look like immunoglobulin domains. So that looks like a decent segmentation. Over here on the left, we have options for how to color this structure representation. The default, which has already been done, is domains. So it will just assign colors based on domains. And the other two buttons, you should use them depending on whether your structure is from the PDB or whether it's from the AlphaFold structure database. If your structure is from the PDB, then the B factor column in the PDB file will correspond to the so-called temperature factors of the experiment. So that would be the more appropriate sort of coloring to use. And you can see that this is quite a well-resolved structure. There's only a couple of tiny deviations from otherwise very good B factor values. If you were using an AlphaFold structure, you would use color it as PLDT, but in this case, the numbers aren't particularly meaningful. And beneath that, you have color keys for the temperature and PLDT ranges. We use the same PLDT ranges listed on the AlphaFold structure database website, and the B factor temperatures are a rough guide to whether these regions of the structure are well resolved or not. The most important part of the results table is the domain assignments region, where we have a listing of the individual domains that have been found, as well as the residue ranges that are covered within each domain, as well as the color used to show each in the domain colored representation. Merizo also has a special property that it won't necessarily assign every residue in the structure to a domain. If it decides that certain residues in the structure should be considered as non-domain residues, then it can do that. And here the residues, the parts of the structure that are marked in gray, are considered non-domain residues, and there's only a few of them in this particular entry. One of the areas in which Merizo really shines is in its ability to deal with these unstructured regions that you find in many AlphaFold database structures. So here's an example that we've taken from the AlphaFold database. If you're interested, this is the UniProt ID associated with it. And you can see that it consists of quite a lot of these unstructured regions that we informally call spaghetti regions. And you can see that Merizo has done a pretty good job of not being distracted by those regions and identifying the three well-structured domains hidden inside. When dealing with AlphaFold structures, it's a particularly good idea to take a look at the coloring based on PLDT. And it's a good idea to make sure that the domains that have been identified match the regions of the structure that have a high PLDT score. So looking at our PLDT key, I can see that the more bluer colors are the regions with high PLDT or the high confidence. And when I switch between the two coloring schemes, I can see that the domains are found in the high PLDT regions of the structure. So this is really a good sign. Finally, let's take a look at a case of protein structure that consists of discontinuous domains. Merizo is absolutely fine with segmenting structures that have discontinuous domains, and these are simply domains in which the residue range that is involved in the domain is not continuous. So let's take a look at this example. This is a structure of pyruvate kinase, and if you're interested, this is the UniProt entry associated with it. Again, this is a structure that I've taken from the AlphaFold protein structure database. So this structure consists of three domains that I've taken a look at, and I think that's a pretty good segmentation. Again, because this is an AlphaFold structure, I want to look at the PLDT scores, and sure enough, my domains are being called in the regions that are most confidently predicted, so that's good. And looking here at the domain assignments, we see that the domain region column looks quite different, and this is because the domains are discontinuous. If we were to follow the protein chain from the N terminus to the C terminus, it would go, if I was to count the domains, it would go domain 1, domain 2, domain 3, domain 2, domain 1. And so if I was to follow the chain within this protein structure, it would kind of travel from the blue domain into the green domain, into the red domain, and then back into the green domain, and then back to the blue domain. So when we have discontinuous domains, we have these underscores in the domain region column. And this simply means that if we're interested in the residues belonging to domain 1, it starts at residue 54 to 85, and then 432 to 574. Similarly, for domain 2, its residues 86 to 159, and then 262 to 431. And the third domain, domain 3, is a continuous domain because it only has one such sort of sub-range within it. So if I was to follow the numbers, I can see that it goes 54 to 85, 86 to 159. So I've now entered domain 2, and then 160 to 261. So I've entered domain 3. And then after 261, 262 is in domain 2. So I've entered domain 2 once again. And after 431, 432 is back in domain 1, so I've had to go back into domain 1. So this is how you would interpret discontinuous sort of domain ranges. Finally, let's take a look at the different output files that you can download from a Merizo job. The downloads are listed here on the right. You can download the result files individually. There's a few files that are generated by a job, or you can download them all together in a single zip file. I've already downloaded this file, and I've got the contents open here in VS Code, but you can use your favorite text editor. And we'll start with this file that has the extension .merizo. This is a file in so-called TSV format, or tab separated values. So I'll just quickly tell VS Code to treat it as such and realign the columns just so that we can see it conveniently. And we'll go through the columns of this file. The first one is simply input, and this just has the input file name that was supplied to Merizo. In this case, it's a UUID that was used to uniquely identify your job on the web server. NRES, which stands for the number of residues in your input structure. NRES DOM, which stands for the number of residues in your input structure that were found to be in structured domains. NRES NDR stands for the number of residues that were found to be in so-called non-domain regions. And these are those regions where Merizo has decided not to assign them to a structured domain. NDOM stands for the number of structured domains that Merizo was able to detect. PIOU stands for predicted intersect over union. And this is essentially a confidence measure for Merizo's segmentation. In the paper, we found that values of 0.75 or greater corresponded to more reliable segmentations, and so this one can be considered a reliable segmentation. The next column has the runtime that Merizo took for this particular segmentation in seconds, and the last column is result, which has the residue ranges that correspond to the different domains. Individual domains are separated by commas in this column, and so you can see that there are 3. This is the segmentation of the pyruvate kinase structure that we were just looking at. And so you can see that the individual residue ranges are shown here for 3 domains. As I mentioned previously, if any of the domains are discontinuous, they'll have these underscore characters in them, but otherwise they'll just be sort of A to B, start to end. So that's quite a useful file to look at. The next file is this .pdb2 file, and this corresponds to essentially the atom records taken from the PDB structure that you provided, Merizo. The only difference is that the occupancy column has been modified to give domain membership information. So where this column is 0, those correspond to the non-domain residues. And then for the domain, the residues that are in domains, you'll find sort of 1, 2, etc., corresponding to the individual domains that were found. This file can be opened in a standard structure viewer such as PyMol, so I'll just do that quickly to show you what it looks like. So we'll take the PDB2 file and open it here. And as you can see, it can be viewed just like any other PDB file. If we want to color this using the occupancy information that we've put into the file, we can type spectrum Q, and this will color it according to the domains. When viewing it in this way, the dark blue regions, the regions corresponding to an occupancy of 0, will appear as dark blue. Otherwise, the structure has been colored according to the structured domains identified by Merizo. Okay, returning to the output files, we'll take a look at this .idx file. And this is essentially a one-to-one mapping. For every single residue in your input structure, what domain does it belong to? And again, if the number on the right-hand side of each colon is a 0, that means that particular residue belongs to a non-domain region. Otherwise, you can see that residue 54 onwards is in domain 1, 67 is still in 1, residue 100 is in domain 2, and so on. So this assigns a domain ID, if you like, to every single residue in your input structure. If your structure is taken from the AlphaFold database, it will almost always, I think always, start with 1. But if it's from the PDB, this will start at the first numbered residue in that PDB structure. And then finally, we have these files that have this DOM-PDB extension. And these are, again, PDB files similar to what we saw previously. They are essentially subsets of the lines in the PDB2 file corresponding to the structured domains. So here you can see that Merizo v201 has 1 in the occupancy column, domain 2 has 2 there, etc. So if you're interested in viewing the structures of these domains in isolation, you can do that too. So these can again be viewed like regular PDB files. And because they have this somewhat non-standard DOM PDB extension, we have to do a little bit of extra work in PyMol. So we just copy the path there and say format equals PDB. And then we can view the structure of the individual segmented domain in isolation. And I think that pretty much covers it. Job configuration.csv simply tells you what version of the software was used. But otherwise, I think that pretty much covers it. Thanks for watching.